Human blood is a highly specialized type of connective tissue, in which cells are suspended and carried in a fluid called plasma. The volume fraction of blood occupied by the cells is defined as the hematocrit, or HCT. The spectral and spatial distribution of light incident on blood not only determine its appearance attributes, such as color and translucency, but also provide crucial information about an individual's health, such as the oxygen content of his blood. The CLBlood model employs a first principles approach that takes into account the specific optical and morphological characteristics of the blood cells, whose biconcave shape is accurately modeled using spline surfaces. Each cell is probabilistically positioned on the fly during the simulations and then discarded. Accordingly, light traveling in the volume can be absorbed within a cell, absorbed by plasma materials, or propagated to the surrounding medium. The multiple reflections of light within a cell are also simulated. For formulation details, please refer to our paper. The model predictions were quantitatively evaluated through comparisons with actual measured data. The shapes of the modeled curves closely agree with the shapes of their measured counterparts. They also showed that as the HCT increases, the reflectance decreases below 600 nanometers and increases above 600 nanometers. The scattering profiles also show a quantitative agreement and demonstrate the strong forward scattering behavior of whole blood. The model predictions were also qualitatively evaluated through comparisons with experimental observations. For example, a reduction of oxygen content results in a reflectance decrease between 600 and 800 nanometers, a peak around 480 nanometers, and a dip around 760 nanometers. For samples with low HCT, the sieve effect can reduce the amount of light absorbed by whole blood in comparison with hemoglobin solutions or hemolyzed blood, notably in bands of pigment absorption maxima. For samples with high HCT, the detour effect can increase the amount of light absorbed by whole blood in comparison with hemoglobin solutions or hemolyzed blood, notably in bands of pigment absorption minima. Motion also affects how light is reflected and transmitted by blood. As the shear rate increases, the cells start to align with the flow direction. This change of orientation increases light reflection and reduces light transmission. We have generated images to further demonstrate the predictive capabilities of the CLBlood model. Here we present images illustrating the dark red appearance of deoxygenated venous blood. And the distinctive bright red appearance of oxygenated arterial blood. This scene displays samples with hematocrit equal to 0.49, 0.35, 0.15, and 0.05. The color of blood may also be affected by the presence of dysfunctional hemoglobins. This sequence depicts methemoglobin fractions equal to 10%, 30%, 70%, and 100%. Similarly, this sequence depicts self-hemoglobin fractions equal to 10%, 30%, 70%, and 100%. Finally, we illustrate the visual effects of thickness variations on the overall appearance of a blood sample. As demonstrated, CLBlood can predictively simulate the spectral and spatial responses of blood under a wide range of physiological and rheological conditions, making it the most comprehensive optical model for blood developed to date.